Welcome to iBiology. My name is Steffen Schmidt, and today I want to introduce the technology of cell sorting to you. So when I'm referring to cell sorting, I mean FACS in its original sense. FACS is the abbreviation for fluorescence-activated cell sorting, and many people say FACS when they just mean flow cytometry, as it's shown here in the cartoon on the right side. So here we have a single cell suspension labeled with fluorescent dyes, which we bring here in front of a focused laser beam and the optics, where we then are able to detect the fluorescent signals and convert cellular structures into light signals. But by far, we are only analyzing those cells and not doing cell sorting. So what are the reasons that we need a cell sorter at the end? If we would apply a laser on our tube of cells, we would not get really important and valuable information out of that. Instead, we want to do many fancy things with our cells, like cultivation, transplantation to look for stemness of our cells, and do all these omics, which are available today, like proteomics and genomics. So for that, we need to do cell sorting, and this was developed in the immunological surrounding. But in the meantime, cell sorting established as a very important technology in the biomedical research, ranging with a lot of different applications, starting with GTL, so-called, where we start separating GFP transduced cells, so green to the left sorting, because we maybe also want to establish cell clones with them. We want to synchronize cells for cell cycle, transplant them. But in the past, very high demand was on single cell analysis, and their single cell transcriptome analysis is getting more and more important. Today, I'm focused on the droplet electrostatic cell sorting technology and all these other possible mechanisms like the magnetic activated cell sorting or chip-based and microfluidic devices will not be discussed in detail today. So now let's have a look on the machine itself, and what we can see that many features are shared with a common flow analyzer. We have a shear stream bringing our cells in front of a laser beam. This is done inside a cuvette. But for sure, we also need additional topics and features like a transducer, a charging device, a nozzle, and deflection plates, enabling us later on to separate the drops from each other. So cell sorting can be thought as a two-step process in principle. In the first step, we are doing our conventional flow cytometric analysis, as is discussed in detail in different sessions. But then we start injecting our stream into the air, break the stream into small droplets, and then on the way down are able to separate these droplets into different positions. So basically, we are doing droplet sorting and not cell sorting, and we are only calculating when a cell of interest is reaching this particular drop. That means that everything, all the particles which are included in the drop, will be sorted out finally. A real picture of an instrument where you can also see that all the components are in a fixed and a precise position. There is one exception. This you can call the heart of the flow cytometer and a cell sorter. This is this nozzle part, which is sitting here. This is flexible, can be taken out, and therefore this allows us to adjust the instrument settings to our experimental needs. This is how modern nozzles look like. Here we have this orifice coming in different diameters, ranging from 50 to 130 micrometer. And for sure, the size of this diameter is later on then influencing very much our stream characteristics. So let's have a closer look how the droplets are really formed in an instrument. Like in a loudspeaker, we have a vibrating membrane, which is later on transferring a stable wave onto our stream. So below the nozzle, we have a camera, so we can observe this droplet formation. And as indicated here on the left, we have here different nozzle sizes and running with different frequencies. That means for each nozzle, we are generating a specific amount per drops per second, which are then finally allow us to adjust the speed of our sorting process. There are different possibilities how to control our droplet break-off. One of that is the amplitude, and again, when you think about a loudspeaker, the audibility of the tone would be comparable here to the amplitude, to the force we are applying to the stream, and with that we can adjust here the drop one formation in a very rough manner. The second thing we can control with the amplitude is the so-called gap, which is the distance between the first free drop and the last attached drop on the stream. And the gap is later on important to get a stable side stream, because the gap is serving as an insulator of the different charges we apply, and only with an optimal gap we have nicely separated side streams later on without spraying and potential contamination later on. So drop one itself, we will hear in a few slides. It's very important that we keep this position quite stable and that we can precisely calculate where this drop one is located. So once we generated our drops, we can charge them, and later on we can deviate them in an electrical field. So we are using physiological salt solutions as a carrier stream, as sheaths. So this sheath stream, sample stream, can be charged, and this charge is then traveling down the stream to the droplet break-off point, and in the moment when the drop is leaving the stream, it also carries the same charge as the stream before. And by going down, it will then move here to an electrical field and is then deflected in the right manner and can then be collected with the right collection device. So now we have to bring all the things together. So we have the point where we deciding if we want to sell or not. This is the laser interrogation point. We have a droplet break-off point, and now we have a charging of this drop, and now all these three parts had to be synchronized, and this can be done in the so-called drop delay, which is basically the time span or the distance a cell needs to move from the interrogation point down to the break-off point. We can accurately calculate this time span because we are running the system with constant velocity, and what we also mentioned already, that the break-off point is very constant. So also the time is constant and can now precisely calculate it. The number we are getting out is not the time in seconds. It's basically really the distance which is fitting inside. So the drop delay is given as numbers which are fitting in the distance between the interrogation point and the droplet break-off point. And in this example would be 6.5 drops. To define the drop delay, there are different systems available. The most prominent one is the so-called Akku drop system. This consists of a separate laser which is equally illuminating the deflected drops and the waste stream. So when we're now using fluorescent beads, we can simulate the sorting process, and when we calculated the right drop for sorting, all the beads should be sorted out and no bead should be left in the waste stream. So this is how it can look in an instrument, in a software-aided system. So we have now two areas, one for the waste and one for the deflected stream. And you see with a correct calculated drop delay, all the fluorescent labeled beads are sorted out and none is left here in the waste stream. So we are looking for a minimum here in the waste stream, and the calculation between sorted and unsorted events should sum up at the end in 100%. There is a method published by a colleague from mine, Andy Riddell and Rui Gardner, which is called RMAX method, which is a quick and independent way to prove your instrument settings beside the AccuDrop system. What you also can see that the actual drop delay values can vary from nozzle size to nozzle size and are actually quite bigger than the one we had in the example. So we now heard about the theory. We now know how to justify if you want to sort or not. Now let's assume a more realistic daily situation. You have an experiment where you need a very pure sample with a maximum of your cells. But as always, time is money and you need it very fast. Of course, all these three things, purity, yield, and time, cannot be maximized at the same time. So therefore, such a request would just not be possible. So to understand this, we first will have a look what are realistic sort speeds and how fast can we go. We already saw that with a smaller nozzle, like a 70 micron nozzle, we can generate more drops. So with 90,000 drops, we can run about 30,000 cells per second. So this is giving us 100 million cells per hour. This sounds quite much at the beginning, but if you now start thinking about rare cell populations, you will end up after a full day of sorting with not more than 2 million cells. If you now have very complex experimental setups, this might already be a limiting cell number to go on. So why do we have to limit to 30,000 cells per second? If I just told you that with a 70 micron nozzle we have 90,000 drops available. And this is because we don't get an equal distribution of our cells inside the stream, like in this ideal situation. Under this situation, we would have no problems to discriminate rare cells and to sort them out very efficiently. Unfortunately, the reality looks a little bit different. So we have a more or less random distribution of the cells inside the stream, and therefore also a quite high possibility that two cells matching up in one drop. And therefore we have some difficulties that rare cells are completely free of unwanted contaminating neighbors. So now let's have a look again on our numbers and what is given by the physics. We already learned that with going down with the nozzle size, we can increase our sort speed. But we have to pay a price for that. To get a stable droplet break-off with a small nozzle, we have to do a high pressure, apply a very high pressure on the stream. This induces shear stress and forces to the cell, and therefore the cell viability will be higher when we are choosing a larger nozzle size. To summarize some of these features, we can come up with some rules of thumb. For example, cell size is mattering, which should not exceed 1/3 of the nozzle diameter. Otherwise, again, the shear stress will become more important and the cells start dying sooner after sorting. And also the side stream will not be as stable as possible. Another thing is the optimal event rate you can run on those machines. and this is roughly 1/3 to 1/4 of the drop frequency. And therefore, also we are coming down to the 30,000 drops per second we can run at maximum. So you see, all these settings together are influencing our decisions and also are dependent on our downstream assay. So for sure, if you want to have a viable cells for transplantation or for cell culturing, then you would choose a more gentle treatment of your cells at the beginning. We heard about these three terms, yield, purity, time, and we can put that here in a kind of triangle because all three terms are influencing each other. And one of the reasons is that you cannot really maximize all of them at the same time. You always have to find a compromise to that. If you have a focus on your yield, then you have to sacrifice your purity or you have to increase the time. The same way, if purity is an issue, then either your yield is going down or again, time does matter. And like in our example, if you have less time, then you have to live with a lower output and also with less purity. Sometimes there is a mixing or misunderstanding in terms. That's why I explain the meaning of yield to you. So let's assume you have a 10% positive cells in your sample and we are analyzing 100,000 cells. That means at the end we had 10,000 cells which could have been sorted out. Now after the sorting process, the machine tells you that you only have 9,000 cells sorted. So the yield is then 90%. 90% of that what you potentially or optimal could have sorted out. But why are we losing now 1,000 events? This is due to the fact, as I said, we have a random distribution and therefore cells are matching up in one drop or coming too close that the purity would be impaired, and this is then called a coincidence or an electronic abort if you run them too fast. So all these effects have a consequence that your yield is normally always lower than what you originally calculated. But even with a yield of 90%, you cannot be sure that you really refine these 9,000 cells in your collection device. What you really find is the recovery of your cells, which is then normally between 70 and 80% of your yield. Why is that? As I said, cell sorting is very stressful to your cells. So they start dying, getting fragmented, or cells are living entities. We are using mostly plastic collection devices, so the cells can stick to your plastic tubes and the walls and therefore can get lost. Also, the sample preparation and processing after the sorting could impair the recovery. Think about the centrifugation. Also, there you can lose some of your cells. Now, it would be very stressful and demanding to find always a good compromise between this yield, time, and purity. And therefore, the machines are giving us already predefined options to choose, which are the so-called sort precision modes. And they are coming here in 3 different flavors. Of course, there are more, but I'm just introducing these 3 to you. And already what the names are suggesting, one or the other term is then prioritized in that. And these precision modes are always a mixture and combination of the so-called sort masks. So now I'm already introducing the next term. So now let's have a closer look what the sort mask is about. Most cell sorters are knowing 3 different sort masks: the yield mask, the purity mask, and the phase mask. So each mask is dividing the drop virtually into 32 segments. And then a cell can be touching or located in one or more of the segments, and this will then influencing the outcome of my sort decision. For example, the yield and the phase masks are looking on the drop we just calculated in the drop delay, whereas the purity mask is looking on the trailing and the leading drop to look if there are contaminating neighboring cells next to our cell of interest. So all these three masks are now combined in these sort precision modes, which I summarized now in the next tab. So from the sort software, you can now choose here yield, purity, and single cell. And if you have chosen the yield mask, you see the yield is set here to 32. So that means a cell will always touching a segment, and therefore always 2 drops are sorted to make sure that the cell of interest really makes it into our collection device. So the aim is to get all of our positives, and therefore we are getting a very high recovery. But because we are sorting 2 drops always, we are also getting a higher volume. So in the yield setup, you see we don't care about the purity. This is coming in the next combination, where we, in addition to the yield mask, we are now adding the purity to maximum, so that we only get the positive ones with a very high purity. But that means if we have a contaminating neighbor somewhere in the attracting drops, that we're also losing cells of interest. The last option is the single cell option, and this is chosen very special when we, for example, think about cloning our cells, where we really want to have single cells in our 96-well plate, for example. There we set the yield mask to 0 because we want to sort only 1 drop. We want to sort with a very high purity, and to make sure that the drop is not getting empty, we're also setting a phase mask to 16. So only a drop is sorted when the cell is located in the middle of a drop, and there's no chance that this cell is moving to the next or to the drop before. So we're getting our cells in very low volume, but for sure we are losing most of our cells of interest because it has to come together that they are located in the middle of the drop without any contamination surrounding. Please be aware, if you set the phase mask to 32, then no drop will be sorted because the cell is then always in the phase mask, and this will lead to 100% abortion of your cells. At the end of the talk, I want to discuss some strategies, how you can improve your sort quality and the outcome of your sorting experiment. As you are used in conventional flow cytometry, you should exclude doublets and dead cells from analysis. You should think about your target collection medium, because this can have an influence on the viability of the sorted cells. If time is an issue, for sure you should think about if you can pre-enrich your sample beforehand. And at the end, I will show you some examples how the setting of threshold and gating strategy might influence your outcome of the sort. No way to comment that you also need to do proper compensation. Even if in conventional flow cytometry you can change your compensation settings afterwards, keep in mind that then at this time you already have sorted your cells, and then you cannot change your sort gates anymore. Let's come to the threshold. So here are 2 examples for differently set thresholds. In one situation we have a very low threshold where all the particles can be seen, whereas in the second situation we have a higher threshold which is then also leading to a higher percentage of the target populations. So the advantages might be obvious, namely that you can analyze your cells faster because rare populations getting visualized better in your plot. So you have an overall higher throughput of sorting, and maybe you're also then therefore getting slightly higher yield of your cells of interest. But please always keep in mind that particles which are below the threshold cannot be seen by the machine and therefore are not analyzed. So your purity will be impaired very significantly because you cannot exclude unwanted events from the sort decision process. To come to an end, we will also have a look on the gating strategy. Here I choose an example for regulatory T cell staining with CD4 and CD25. So you can see two populations, one CD4 single positives and one CD4, CD25 double positives. And for both populations, we set now two sorting gates for a high and a low expression of these markers. And if we now have a look on the reanalysis after sorting, we can clearly see that here with the CD4, CD25 double positive cells, we are getting here a good signal. And therefore, we are able to separate P2 and P3 very nicely. Whereas in the CD25 negative population, P5 and P4 cannot really distinguish from each other because here we are in the background, and if you reanalyze the background, it always gives you the background back. So this is just due to the data spread inherent to the measurement and has nothing to do with being more positive or more negative. When you are performing cell sorting, always ask some questions beforehand so that you can then from those things conclude what kind of instrument setups, what kind of nozzles and sort precision modes you are choosing. What is your downstream assay? Do I need cell viability? What is the desired number of cells I'm having to do so? What is inside my cell? Can I reach these desired cell numbers? What is the percentage of my target cell? Is there maybe a high chance for coincidence to do so? Therefore, which parameter do I need to maximize? Is it purity, yield, or is it time? Because my instrument is heavily booked and is not accessible all the time. Can I do pre-enrichment? Or what is my target collection device? So many things can be clarified beforehand before you even go on the machine and starting your process. If you're now getting interested in this technology, I can recommend some books and publications to you for further information. So there are two books. One is the Howard Shapiro, The Practical Flow Cytometry. Howard is a famous teacher at the moment here in flow cytometry, and his book is dealing a lot about flow cytometry at all, whereas the book of Andreas Radbruch is also focusing a little bit more on cell sorting. And quite recently, there was a publication in the European Journal of Immunology where some colleagues and I put some stuff together to give you a good starting point for flow cytometry and cell sorting at all. But at the end, please keep in mind we are always dealing with living cells. We're doing very harsh things to them and try to treat them as gentle as possible. Thank you for your interest.